Let's see. So I have a couple of announcements. One is, oh, go ahead.
I have a question about the exam.
Yes.
Is it all like a take-home portion, or is there going to be some in-class?
It is entirely take-home. Yes. Yeah, you bumped into one of my announcements. I'm going to remind you all. Yes, there is an exam. I will get it to you. I will post it on Canvas on Wednesday. It is a take-home exam. Yes, do the whole thing right there in your comfortable place of work, and it'll be due Friday night. Yes.
Is that is it like a Canvas quiz, like where we open it once and we have to complete it all in that segment? Or
Yes.
Okay.
Yeah. There'll be a few. There's going to be a couple of essay questions in there that I'll have to grade, not Canvas, but otherwise, yeah, it'll be just like the problem sets I've given so far.
Okay.
Just longer. Yes.
So I don't know if I was the only one that didn't know that we had a quiz due until you emailed us. So do we have a weekly quiz every single week?
That's going to be every week, yeah. Except this week. I figure if I give you an exam, that's enough.
So an exam means there won't be one?
Correct. Yeah. Or did you want one?
No. Thank you. So sweet for asking.
Yeah. Yeah, I figure we'll do the exam and then you can get it done and turn it in Friday evening and just go relax for the weekend. That always works. Yeah. Okay. The other thing I was going to mention is the lab this week. As you know, again, what I'll do every week, there's a little video explaining what you're going to do in the lab. The lab this week starts off easy, but it's going to demand that you come in at times outside the scheduled lab hours. Yeah, so that's not so good, but you know, the flies rule everything right now, so you just have to obey them. You should go in and check before your lab time and see how they're doing. I was up there this morning admiring your babies. They're working. There's little maggots all over the place, which is what we like to see. Okay, let's see. Was there anything else I needed to tell you? Any questions? Yeah, if you have questions about the exam, I will post it before class on Wednesday, so you should take a look at it at that time. And if you have questions about the basic exam, yeah, you can ask those in class too. So I'll set aside some time for that. Yes?
So the exam is due on Friday?
At midnight, yeah. Yeah. 2 days is enough, right? Yeah. Okay. So where we were, let's see, where did we leave off? I've been talking about a couple of things. So 2 weeks ago, I covered a little bit of the history of genetics, and I was telling you about in the 19th century, everyone was floundering. They didn't have a good theoretical explanation for how genetics works, which of course is no barrier to the pragmatic business of doing selective breeding and hybridization, all that kind of stuff they're doing. But they didn't know how it works. And I told you that Darwin came up with his little hypothesis, his gemular hypothesis, which also didn't work very well. So yeah, you remember that. We've got these things called gemmules that are supposed to congregate in the gonads for fertilization. And they're found in every part of the organism. There are specific types for each of the organs and tissues. They're migratory. All these details. They're fused during development to specify the parts of the body. And for evolutionary theory, this is kind of important. They're capable of modification in the adult. So we have inheritance of acquired characters. And Darwin also proposed blending inheritance. That children were kind of a blend, a mix of the two attributes from mom and dad, etc. Okay, this hung around for a long time, about 30 years. Darwin proposed this in the 1870s, and lots of people were unsatisfied with it. For example, this is the work of Francis Galton. Francis Galton was Darwin's cousin. And he had the great, let's go test this gemular hypothesis. And one way we can do this is with this cruel experiment, where you take a black rabbit and a silver gray rabbit, and we ought to do a transfusion of all the blood from this black rabbit to this one. It's kind of a horrible experiment to contemplate, because you basically plug a tube into this rabbit and stick it into this rabbit, and another tube that drains this rabbit, and you basically transfuse all the blood from this poor rabbit to this one and get rid of its existing blood. And then you have rabbit for dinner. Lots of rabbit. Okay, so if the gamular hypothesis was true, you think, hey, well, we could shuffle around some of these gamules by these transfusions and transfer characteristics like coat color from one organism to another. Galton did this commendably with the support of Darwin, who actually provided the facilities to do this, and it didn't work. Silver gray rabbits still gave birth to silver gray rabbits. Darwin kind of shrugged it off and said, well, maybe they're not transporting the blood, maybe they're transporting the lymph. That's kind of a bad sign. We have to be revising your theory that much in order to make it work. Okay, but what really sealed the deal for Darwin was this. This is a Scottish engineer named Fleming Jenkins. And Fleming Jenkins did not much care for the implications of Darwin's theory of inheritance. And he wrote a letter to the newspaper, and he argued against Darwin's whole model of genetics. And this is the argument he made. And he said, okay, suppose a white man is wrecked on an island inhabited by Negroes. Uh-oh, this is Victorian England. There should be little alerts going off in your brain. There's going to be a problem coming here. Anyway, he says, okay, suppose you got a shipwrecked white man, Tarzan. And he joins up with a friendly tribe of dark-skinned people. And then, of course, suppose him to possess the physical strength, energy, and ability of a dominant white race. There's an assumption jumping out at you. And grant him every advantage. And then, no matter what happens in the struggle for existence, he's going to be breeding with the native population, and every generation will dilute out his beautiful whiteness. And according to Darwin's theory of inheritance, it would just disappear over time. Yeah, you can see there's a whole bunch of racist assumptions in this. But the thing is, maybe it's kind of an indictment of Darwin. This argument really hit him hard. He kind of dropped the whole idea of blending inheritance after this letter. Because, it's kind of striking at his prejudices and saying, if your model is correct, then all this stuff you believe about the superiority of the white race is going to fail. Huh. Okay, so, and this is, and actually, although it's couched in rather racist terms, this is a valid argument. Yeah, if you get a model of inheritance that's just blending, then every generation is going to dilute whatever attribute gave you a benefit in the first place. Okay. So, here's Darwin. He's got this theory out there. And it's got a whole bunch of problems. Here's a little list of all the problems that were coming up with his theory. So, first of all, we never see gemmules. People are using those fancy microscopes, and they're getting in there, and they're looking really hard, not seeing anything that behaves like what a gemmule should do. So gemmules were something that Darwin just kind of invented as a hypothetical. You didn't see inheritance of acquired characters. Yeah, we just didn't see that happening. Remember that example of the Jewish doctors who were telling them, yeah, we've been lopping off foreskins for thousands of years. Jewish babies are still born with foreskins. So that's a problem. Fleming Jenkins' argument. So blending inheritance was incompatible with what we see. That, traits pop up in odd ways, generation after generation, and that's also going to be incompatible with evolution. Darwin's baby. So his model of inheritance was in conflict with his theory of evolution. And then here's the big ones. As I told you last week, there were all these observations people were making of cells, and they were identifying these chromosomes. Chromosomes are cool. Chromosomes must have something to do with this process of inheritance. They're not accounted for in Darwin's theory. There's also this idea by Weismann, Weismann's barrier. Yeah, it's central to Darwin's idea that gametes are moving from all the somatic tissues into the gonads. And Weismann is just saying, nope, that doesn't happen. You don't see that. And then in his response to Galton's criticism of his model, it's an ad hoc hypothesis. It's not built on a foundation of good, solid evidence. So you can just sort of fudge it as you go. And that's never a good sign for a theory. Okay, so he's proposed this in the 1870s. It's going to hang around until the 1900s. And why? Why didn't people just say, wait a minute, this makes no sense. Let's throw it out. There's a couple reasons that I list here. One is that Darwin was very influential. He was the big name scientist of the Darwinian area. So if you criticize Darwin, you better have all your ducks in a row, because everyone's going to jump on you. So people are really reluctant to oppose it too hard. And then the other thing is nobody had a better explanation. Well, great, you don't like Darwin's idea? What are you going to replace it with? So we're just kind of hanging around in limbo there. People are still doing all kinds of breeding experiments. Lots of people are studying this. It's still an active problem, but they don't have that nice, integrated, complete, consistent theory to explain this. And this, I think, is an important word: sociology of science. Yeah, we like to pretend that science is all objective truth, right? But as we can see, for instance, in Fleming Jenkins' objection, it's also colored by social biases. And it's not always as cut and dried as we'd like it to be. We're just constantly striving towards the truth. We don't have the truth. Okay, so we're going to have to get rid of this theory. Somebody better come up with a better idea than gametes. And that's where this guy comes in. You all heard of Gregory Mendel, right? Yeah, how can you avoid it? Basically, this is Mendel's model of genetics is basically all that's taught in the high schools, I've noticed. We're going to get beyond it, but most of you have probably been exposed to this model. Anyway, Gregory Mendel was a really interesting guy. So he was living in what is now Czechoslovakia. Back when he was born, it was part of the Austro-Hungarian Empire. But he's born in 1822, so he's a little younger than Darwin. Okay, a dozen years younger than Darwin. In his youth, he was an avid gardener and beekeeper. He just loved that nature stuff. And then he had a calling. And he felt like he was supposed to go off and do things for the Lord. And so he became an active member of the Catholic Church. He joined the Order of St. Augustine as a monk. And partly it was because, as from all we can tell, reading his works, he had a sincere call. He really felt that this was an important part of his life. But the other part of it was, he's a sciencey nerd. He was living on a farm. His father was kind of disappointed because his father hoped that he would take over the farm when he was of age. But if he joined this monastery, he could get a free education. He could get a free higher education. They would send him off to universities, and he could study. So it was a smart move on his part. So he goes off to a couple of universities in the region. He's a student primarily of mathematics and physics. You know, one of the reasons the monastery sends you off to do this stuff is to get training so you can be a teacher in your community. And they needed a science teacher. Let's go send Gregor off to study math and physics and all that important stuff. He also studied animal breeding. He worked with animal breeders in there. So he's kind of, that was the thing back then. You didn't get too focused early on. You got breadth of experiments. So he studies this stuff. He apparently was not a great student. He failed his oral exams to get his diploma twice. I don't think he ever got them, the diplomas. But he knew the stuff. He was good enough to be a teacher. So he comes back to the monastery, and he is going to work as an experimental geneticist in his spare time. He's got other things to do, right? He's a priest working in a community in Brunn, in Czechoslovakia. He's got jobs to do. So in his spare time, he's raising plants, raising pea plants in particular, and doing experiments with them. He was good enough at his job. Okay, so he was doing the pea experiments in the 1860s, roughly. And he was good enough at his job that towards the end of that time, I think it was 1868, he becomes the abbot of the monastery. So lots of responsibilities. He kind of gives up science at that point. He wasn't getting much attention for it anyway. But he had done this work, this really interesting work, and he published it. It just kind of sank without a trace. And then he became a professional priest, basically. I think people liked him. He did a good job. So as near as we can tell, he was happy in his life. So, yeah, do that. It's follow your bliss, as we say. He died in 1884, just a few years after Darwin. So he and Darwin really overlapped. They were working at roughly the same time. They were definitely contemporaries. But there were differences in their life, of course. Mendel is kind of an amateur. He's not a world-famous scientist like Darwin. But he's going to do it anyway. Okay, so he does all these experiments with pea plants. And what was he doing? Let's see. Who here has done gardening? Yeah, some of you. How many of you have grandparents or parents who are into gardening? Yeah, I'm not so much of a gardener, but my wife enjoys it, so she does stuff out there. And you all know, you can get these really cool seed catalogs. This is true now. It was also true back in the mid-19th century. In fact, they were really important here in the Midwest for disseminating information about crops and giving people access to new varieties and all that kind of cool stuff. So these are some old-timey seed catalogs. This is a contemporary one over there. So the nice thing about this is you could pick up a seed catalog, or you could go downtown to the seed and feed store, and you could browse, and you would find all these different varieties. Yeah, so Mendel's going to be working with stuff that other people had generated by selective breeding and hybridization and all that cool stuff, by collecting them in nature. So he had a huge variety of different kinds of plants to work with. As I said, I've never been much into gardening, but I have to acknowledge a couple of my teachers because I worked my way through high school and my first year of college as a nurseryman, working in a nursery under Taki Nagasawa and Haru Nagasawa in Kent, Washington. And my happy place there, yes, it mentions a happy place. My happy place there was at lunchtime, I could just go wandering through the greenhouses in this nursery. Oh man, it's a wonderful thing to do. You can just, they got higher oxygen levels. Has anyone done this? Go visit a greenhouse sometime. Just stroll through. Yeah, we got one out here. You can just stroll through it. It's wonderful. And you can see all these different varieties. Taki and Haru also raised bonsai, so there's a lot of bonsai. You could look at lots of cool stuff. So, I acquired a little bit of an interest in biology just from that. Although, to be honest, my education there was, I'd come in at 7 o'clock in the morning and Taki would tell me, there's 10 acres of connect that need to be weeded. Get out there and bend over and start pulling weeds all day long. That was my kind of work. Okay, so we got this great resource, and we still have it. Who has visited the horticulture garden? Some of you, oh good, yeah. Yeah, we go that now it's not a good time. It's kind of boring because it's just under a couple feet of snow. But come the spring, if you haven't been there, take a walk. It's about half a mile out past Cougar Stadium. Visit it. It's a gorgeous place. And the focus there is, here are all these different varieties of plants that we grow here. Here's the ones that thrive in the Minnesota climate, etc. And so you can just browse through there and see all this gorgeous flowers and all kinds of plants to play with. Okay, so this is Mendel's situation. He's got it relatively easy at this point. Let's go get some seeds. We can get to the seed catalogs. We can talk to the local farmers. They'll tell you, oh yeah, you want yellow flowers or you want red flowers? Here, take your pick. Okay. So what did Mendel do? This is the big difference between Mendel and Darwin. Mendel is going to seriously practice this idea of experimental reductionism. Pea plants are complicated, right? Lots of stuff going on in a pea plant. So what he was doing is consciously saying, okay, there's all this cool stuff. I'm just going to look at one thing at a time. And I'm going to do experiments on that one thing. I will work out what's going on with this one thing. And only then will I move on to a second thing. And after I finish that one, I'll go do a third. And then later, I'm going to get fancy and just start combining thing 1 and thing 2 and ask what happens. So lots of stuff going on here. But he's doing it in a very methodical, very precise sort of way. This is why we admire Mendel now, because he did things so thoroughly and so carefully. Okay, so for example, he's got these traits that he can find. Yeah, again, go to the seed catalog. There they are. Go down to Willie's right now, the grocery store, and you can find bags of yellow dried peas and green dried peas. It's really easy to get these kind of varieties. So up there at the top, you see, okay, we're going to look at round and wrinkled seeds, and we're going to compare those. We're going to cross them and see what happens. He's going to look at yellow and green seeds. He's going to look at the shape of the pods, the color of the pods, where the flowers are on the plant, the color of the flowers, and the height of the flowers. So he's going to pick 7 things, just 7 things, and he's going to study them into the ground over a number of years, and he's going to get an answer for these things. So this is exactly what Darwin needed to do. Remember Darwin collecting anecdotes all over the place? Man, he could have made a great leap forward, equal to what he did with evolution, if he just stopped, flitting about and said, okay, I'm just going to raise this one thing and see what it does, like Mendel does. Okay, so he's going to do a series of crosses with these guys, one at a time. This is important. He's not going to say, okay, well, I'm going to cross violet flowers with round seeds. Not yet. First, he's got to work out how round seeds and violet flowers are inherited. Okay, what he's going to do is a series of monohybrid crosses. So that's one in which one character is studied by crossing two parents, each of which has a different form of the character. So round and wrinkled seeds, for instance. He's going to get a plant that makes round seeds, and he's going to get a plant that has wrinkled seeds, and he's going to cross those, and he's just going to look at the shape of the seeds in the progeny. And he's going to put on a back shelf the idea of what color flowers they have, how tall are the flowers. Everything else just gets dismissed. We're going to have a laser focus on one attribute. So monohybrid crosses, we only deal with one particular variant at a time. Okay, he's going to do these. He's also going to make reciprocal crosses. His reciprocal crosses don't do anything special. We'll talk about these later. But a reciprocal cross is where you take the parents and you swap them around. So you got a parent with round seeds, you got a parent with wrinkled seeds, and you take pollen from the wrinkled seeded plant, fertilize the round seeded plant. But then you also do the experiment where you take pollen from the round seeded plant and you fertilize the wrinkled seeded plants. So this is just looking to see if there's an effect of sex on it. Is one sex better at transmitting traits to the next one? Well, here's how you test it. You just vary the parents. In all of Mendel's experiments, it made no difference at all. And this is one of the things we got to commend Mendel for as well, is he kept doing them. Yeah, all 7 of those attributes. He tested all of them with reciprocal crosses. Nothing ever happened. But he got the data. Sometimes that's important. You just show, hey, this is consistent. Males and females contribute equally to the progeny. We will see cases later on where that's not the case, but here in Mendel's, that's the way it worked. Okay, so he's doing monohybrid crosses, just looking at one trait at a time. So here's our example. Here we've got the round and the wrinkled peas. I made a little cartoon. That one's round, that one's wrinkled, if you can't tell. We have a couple of terms we get from this. One is the parental generation. The parental generation are the true breeding plants that you start with. Okay, so you're starting with true breeding round seeded plants. That's all they ever do is they make round seeds if you cross them themselves. And true breeding wrinkled plants. And we're going to bring them together and cross those two together. So that's our parental generation. You guys right now are raising a parental generation of flies in the lab, right? So you've got scarlet flies, you've got brown-eyed flies. And they're true breeding. We hope you're going to get results from those, and the next part of the experiment is you're going to cross scarlet with brown flies. So the parents of the parental generation, the next cross, we're going to see the result of the F1s. Okay, so we're going to cross scarlet by brown, or in this case, cross round by wrinkled. Now, jumping ahead, this cartoon jumps ahead a little bit because we're going to see that there are 2 factors in each of these plants. And pure breeding ones have 2 pure round factors in their pea plants, and the wrinkled ones have 2 pure wrinkled factors in their plant, in their progeny. So they're going to pass those on, they're going to mix those up, and we're going to get a first filio, or F1 generation, which is a mix of those 2. Successful. I got big W, little w here. So we're going to mix those up, and we're going to get a hybrid generation in the F1s. So Mendel did this, and what does he see right away? Well, he sees that wrinkle disappears. In the F1 generation, it's gone. Where did it go? This is one of the reasons why he's going to propose that there are two factors, is because he thinks, oh, the wrinkled factor is hiding in the F1s. You can't see it because it's recessive, he says. And the roundness is dominant. This is all coming back to you from high school biology, right? Yeah, pretty familiar. Okay, but then what you do is you take these F1s, and you grow them up and you cross them. And then you look at their seeds, and what do you get? So that's what we're seeing here. Up there is the F1s, the hybrids, that are all round. And we cross those, and we get an F2, a second filial generation. So again, what we're doing in the lab, you got the parentals right now. We're going to cross them. We're going to produce an F1, a hybrid of scarlet and brown. What color do you think their eyes will be? Any guesses?
Scarlet.
You think scarlet? You think scarlet is dominant? Okay. Just to warn you, it's not going to be that way. We're going to see some surprises in the cross that Mendel did not, and that's okay. Dominant and recessive aren't appropriate in this case, but we'll discuss that in more detail next week. Okay, but in the case of these peas, you got this nice result. And he looks and he says, oh, look, there's 1, 2, 3 wrinkled ones here, and there's 1, 2, 3, 4, 5, 6, 7, 8, 9 round ones. We see a 3-to-1 ratio. And you were saying with only 12 plants, you don't have any statistical power. So obviously he did many more than 12. But you get the idea. He sees a 3-to-1 ratio emerging here. Now that's really interesting. Because Mendel was prepared for this. Remember, he was taking all those math and physics courses. He was getting training in probability, understanding how probability works. So he looks at this result and he jumps to a bunch of ideas. He comes up with rules. And these rules have held up pretty well. So just from that one experiment, he's going to come up with a couple of ideas about how inheritance works. And then he's going to test it with all those other traits that he looked at. So, you know, seed color and seed shape and all that kind, and pod shape and plant height and all that. So he's going to test that and confirm this. So he comes up with his first postulate, which is genetic factors are controlled by unit factors. That's what he called them, unit factors. He didn't have any of this allele and gene terminology yet. They're unit factors. They exist in pairs in individual organisms. How did he jump to that conclusion? Because he was familiar with the thing you did in the first week in this class. You know, remember flipping coins? And we got all these combinations. And we basically asked, okay, what's the probability of getting one or more heads when you flipped 2 coins? And we figured out, oh, it's 75%. And the probability of getting just tail and tail, that's 25%. That's a 3-to-1 ratio. So he sees that 3-to-1 ratio and he's saying, oh, there must be 2 things that are getting flipped when we do this cross. Pretty reasonable. Okay, so that's his first postulate. And his second postulate, you're all familiar with. How do you explain how some things get hidden in the F1 generation? Well, one of the factors has to be dominant, and the other is recessive. So like in our coin flip example, we said heads is dominant. Anytime you get a head, you score both of them as heads. That's what we're seeing here. Okay. So he comes up with this rule. There's got to be dominant and recessive traits. The recessive ones disappear in the F1 generation, but they're still there. So they can reemerge in the second generation. Okay, so that's cool. That's a nice rule. And here's his third rule. This one kind of was essential for interpreting the other two. It says during the formation of gametes, paired unit factors separate. We're going to split them apart. And they go into the gametes randomly, purely randomly, so that each gamete is likely to receive one or the other with equal likelihood. Okay, so we get two factors. We're going to split them up to make gametes. So each gamete only gets one, and it's equally likely which one of the two they get. So if you've got round and wrinkled, and they're in the hybrid together, and you make gametes, gametes will get either round or they'll get wrinkled, and there's a 50-50 chance of which one you get. And again, you know this because of the 3-to-1 ratio. If there was a bias, if we were flipping an unfair coin, right, you wouldn't get that 3-to-1. You get some other number. But if you're getting 3-to-1, that sort of says, yeah, it's probably just simply random which one goes where. Okay, so he's working that out. This is kind of an important insight as well. So we're going to see random assortment of the alleles to the next generation. And this 50-50 ratio is what produces the 3-to-1 ratio. Okay. I have a little note here. Warning, later we are going to see cases where that equal likelihood expectation is violated. And that's okay. That's actually kind of important too. Because what Mendel is doing is laying down, here's the basic rules. And if you see that something is not following the basic rules, there's got to be something else interesting going on here. That you're going to have to examine it more carefully to figure out what's influencing the frequency that you're getting, if it's not this 3-to-1 that you see here. We'll see examples of that later in this course too. Lots of examples, actually. Okay. So, like I said, he did this. There's round and wrinkled first. He does that. He does all these experiments. He doesn't get 9 and 3. Look at the numbers he gets there. 5,474 round to 1,850 wrinkled. And you divide those, you get a ratio of 2.96 to 1. Pretty darn close to 3-to-1, isn't it? Then he goes on to each of the other ones, the other traits he's looking at, each of the other 7. So we're going to look at yellow, green, look at there. Yellow and green, when you cross them, the F1s you see, they're all yellow. What's dominant, yellow or green? Yellow, yeah. Yellow is the one that you see in the F1s, in the hybrids. And then you cross those and you get the F2, you get 6,022 yellow, 2001 green. That is a ratio of 3.01 to 1. Really close to 3-to-1 again. And he does this with all of these traits. These are the actual numbers from his paper that he published on this. So you can actually see that data going on. And look, it's awfully consistent. It keeps turning up about 3-to-1. 3-to-1, 3-to-1, 3-to-1. You see that? Oh, that's a simple Mendelian inheritance of a single trait. So get used to that. You see that 3-to-1 all the time. Okay, I've got to say one thing about this is that he published this work, and then years later, when it's rediscovered, some statisticians take a look at this and they say, hey, that is suspiciously good, right? How can you get such good numbers? That's so close to 3-to-1. Did Mendel cheat?
And so there are papers on this subject, just these careful statistical analyses of these. And I say forget it. I kind of suspect that maybe what happened here is Mendel did a lot of experiments. He didn't report on all of them, but he just reported on the ones that best fit, which is kind of cheating, kind of. It's pretty dodgy. Don't you guys do that? You know, if you publish something, we want you to publish all the results so people can interpret them. But he's trying to make a case for his model, and so he throws this out there in the single paper. Yeah, there haven't been any retractions of that paper, fortunately. And people have repeated these experiments and seen that, yeah, they do come out at a 3-to-1 ratio. And we found examples in other organisms. Yeah, we get a 3-to-1 ratio pretty consistently. Okay, I'm going to mention Punnett Squares. And I kind of go against the mainstream here. I really don't like Punnett Squares. Punnett Squares are a tool to help you visualize what's going on in a cross. The problem with Punnett Squares is they only work for the simplest possible crosses. And we're going to be moving beyond these simple monohybrid crosses on Wednesday. So yeah, this will do you a lot of good in 15 minutes, and then we're done. You'll never want to use Punnett Squares again. I always see every year when we do paper exams, I see people scrawling Punnett Squares all over the place. I just want to tell them, no, don't do that. That is the most inefficient possible way to solve a genetics problem. But if you're struggling to visualize what's going on, then they can help. Okay, so this is a Punnett Square. And all it is we take the gametes from one individual. So here we got an individual that's big G, little g. The kinds of gametes that will produce are big G or little g. And we put them on our square up there. And then there is another one. These could be polyhybrids, for instance. And here's another one. These could be ova. This one is also big G, little g. So this is like an F1 cross, right? Take our F1 and cross. And we put these on the other side of the box, and then we just figure out what the intersection is going to be. So this square is going to be filled with big G, little g. This one is going to be big G, as you see there. Little g, right there. And little g, big G here. And we can see all the combinations. So we can sort of visualize what's going on here. And then you realize, okay, little g is recessive. So it's going to be our green one, and all the others are going to be yellow. Hey, this makes sense. We can also see the distribution of the genotypes. I'm going to define all that in a moment. There's one big G to two big G, little g, to one little g, little g. So if you like Punnett Squares, if they help you think it through, go ahead and use them. But they really do become useless as soon as you get to dihybrids and trihybrids. The square just grows. You get an exponential increase in the numbers of squares inside of it, and you'll get lost really fast. I will promise, I will show you a better way to solve these problems on Wednesday. Okay, let's go back and talk about definitions a little bit, because I warn you, from here on out in this course, I start speaking geneticsese. It really is a different kind of language with a whole bunch of assumptions and its own vocabulary, and it is really easy to get lost in it until you get familiar with it, and then it's a nice, convenient, easy way to master the subject. So you got to know these terms. Yeah, I'm prone to do things on an exam like say, oh, we do a test cross with such and such and such and such. And you better know what a test cross is. Okay? Because I won't tell you in the exam. All right, you don't have to worry about this first exam. I'll try to spell everything out this time around. Okay, so we got some terms here. One we've used a couple of times here, monohybrid cross. And a monohybrid cross just involves alleles at a single locus. I haven't defined alleles yet, have I? But I think that's the next slide. So put a pin in that. We'll get back to it. A test cross is a specific kind of cross, in which case we've got an unknown genotype, an individual with an unknown genotype. And we cross it to an individual that is homozygous recessive. There's nothing but the recessive alleles, and that allows us to determine the genotype. For example, you've got round and wrinkled. We just talked about those, right? If you've got a round pea, and you don't know who its parents were, what kind of alleles are present in it? And you don't know. Because it could be big G, or no, I was talking wrinkled and round. It could be big W, little w, or it could be big W, big W. Either way, how do you figure out what it is? Well, you do a test cross, and you cross it to an individual that's little w, little w. And that tells you what kind of genotype you've got. And I'll show you an example of that in a moment. Another common kind of experiment is a back cross. You do a cross between two parental types, and you get progeny. And then you take that progeny and you cross it to one of the parents to see what will happen. Yeah, it sounds very incestuous, don't worry. And we're also going to do that. We're going to be doing the back cross later in the course. It's the easiest way to do a particular kind of cross. We've already mentioned a reciprocal cross. That's where you juggle the genotypes of parents. Just figure out just rearrange them and see if it does anything. And for the first week or two of the class, next week or two of the class, that's not going to make any difference. We'll just kind of nod and say, yeah, we were consistent. We did the reciprocal cross, had no effect. Later we'll see some cases where it does have a dramatic effect. And then finally, I throw in complementation cross. Don't worry about it much. Just to fill you in, you are doing a complementation cross experiment in the lab. You didn't even know it, did you? I will explain that in detail in the lab video for next week. So what is a complementation cross? It's to determine whether two traits you're looking at are allelic or not. Are these variants of the same gene, or are they different genes that work together? I know that's kind of complicated, but trust me, we'll get to it next week, and it won't be on the exam this week. Okay, just want you to be aware of that term. Here's a few other terms. Oh, gene. Gene is a hairy one. People are still arguing about exactly what a gene is. We talked a bit about it in cell biology. Here in genetics, gene has a different sort of meaning than it does to molecular biologists. Yeah, get geneticists and molecular biologists sitting there talking about genes, and you can get all kinds of confusion going. A gene is a unit of heredity whose existence can be confirmed by allelic variants. Okay, what does that mean? That means you get different forms. You know, we could say, oh, there's a gene for seed shape. And there are allelic variants like round and wrinkled. Maybe there are others out there. So we'd say that there's a gene for seed shape. We're going to see later that sometimes there's not a gene. There's multiple genes that work together. That's where that complementation cross is going to come in. So we've got these factors that we can confer by allelic variants. For example, there are some things that we can pretty well assume are genes, if you're a molecular biologist, but a geneticist wouldn't recognize them. For example, is your number of ears genetically controlled? How many ears have you got? You've all got 2 ears, right? You could look across the animal kingdom and you find just about everything has 2 ears. And you might say, okay, well, there's got to be genes somewhere that are specifying the number of ears. But if we don't have an allelic variant, that is, if we don't have a mutant that makes 1-eared people, it doesn't exist to a geneticist. It just, as soon as we find those variants, we can start doing crosses, and we say, oh, there's a gene that specifies how many ears you should have. But there doesn't seem to be. I know, it's confusing. Genetics is a whole different discipline, and geneticists think in some different ways. Okay, another term we'll use that I will tend to throw around is locus. At the level we're at in this class, we're going to sort of treat locus and gene as synonymous. But they're not really. A locus is a specific location on a chromosome. Okay, that location will often fall right on a gene. And so we get a variant in locus that will affect that gene. Okay. But there are things that you can find a locus on the chromosome that isn't associated with a gene, for example. It's going to be invisible to geneticists, because geneticists tend to be blind that way, but it will have an empirical molecular reality that we just don't detect. So here I will kind of use locus and gene interchangeably. So there is a round locus in pea plants. And I could also say there's a round gene, a seed-shaped gene in pea plants. Okay, another important term, phenotype. The observable properties of an organism that are genetically controlled. All right, so in our previous example of round and wrinkled pea plants, okay, round is a phenotype. You can say that pea looks round or that pea looks wrinkled. So we're talking about how it's expressed in the plant. Okay, so I could look around this room and I could say, oh, you have dark hair, you have light hair, you have brown hair, you have green eyes, you have blue eyes, you have brown eyes. All these things are descriptions of your phenotype, what characters you are expressing. The complement that is the genotype. So genotype refers to the specific combination of alleles present in the organism. So an allele is one of the possible mutational states of a gene. It's distinguished from other alleles by its phenotypic effects. It says here Mendel called these alternative forms. So for seed shape, for example, we have round and we have wrinkled. And that's a different description than what we see in the phenotype. We're saying there's some invisible unit factor that makes the seed round or wrinkled, and that these organisms, these diploid organisms, have pairs of these things. So if I've got a round pea, I would say the phenotype is round, okay? And if I want to describe the phenotype, I would have to say what alleles are present. Oh, it has the wrinkled form of the allele of the locus plus the round form. Or it's homozygous, it's got the same alleles present. So one of the giveaways here is, if I ask you about this on an exam about a diploid organism, you can describe the phenotype with one word. Oh, it's round. In order to describe the genotype, you have to specify 2 things. What are the 2 alleles that are present? It could get hairier if I start talking about triploid organisms. I can specify 3. But for now, we'll just settle with 2. So just remember that. Whenever I ask you a question to give me the genotype of this organism, you better tell me 2 things. not just one, or it won't work. Okay, we'll get more into these, and more details on this later. Okay, one other set of definitions that I've already been casually throwing around. Homozygous, heterozygous, and hemizygous. So homozygous, this is a description of the genotype. So an organism that possesses two identical alleles at a given locus is a homozygote. It's got the same two alleles. So if I ask you to give me the genotype of a round pea, you could say it's big W, right? Two alleles. Or you could say it's homozygous for big W, same thing. Heterozygous means there's two forms, two alleles at the locus. So if with my imaginary round pea that I keep holding, it's gigantic, look at how big it is. Anyway, there's this imaginary round pea, and I ask you the genotype. And if it's got a big W and a little w, you could just simply say, oh, it's heterozygous for seed shape. That also describes the genotype. See what I'm saying? We're going to get bogged down in the language here, aren't we? There's a whole bunch of terms that we'll take for granted by the end of the semester. Okay, hemizygote. That's an organism possessing only one allele at a given locus. Can anyone think of any cases where that's pretty common? It has something to do with sex. You guys must know something about it then, right? So all the boys in this room, let's see, there's 1, 2, 3, we're outnumbered. We are hemizygous for all the genes located on the X chromosome. We've only got one X chromosome, then we get this runty little Y chromosome. So we've only got one copy of those. You can't say that we're homozygous or heterozygous. Those make no sense in this case. We're hemizygous, just for the ones on the X chromosome. Women, on the other hand, you got 2 X chromosomes, so you could be heterozygous or homozygous. So an example of that is colorblindness. So that's a gene that's located on the X chromosome, a mutant gene that knocks out one of the color wavelengths that we can detect. So a colorblind man is going to be hemizygous for the mutant variant, and a woman can either be homozygous for the wild-type variant, homozygous for the mutant form, in which case she's colorblind, and that's rare, or heterozygous. Trust me, you'll be throwing these terms around casually pretty soon, right? You're all pretty comfortable with it so far? You've probably encountered these terms elsewhere. Okay. So I got time, very little time, but we'll talk about a test cross real quick. So the test cross is something that Mendel developed. It's to determine the genotype of the organism. So we don't know. It could be heterozygous or it could be homozygous. So, I just use the example of colorblindness. You ladies out there, you got 2 X chromosomes. I presume you all have normal color vision. How do I figure out if you're carrying the colorblind, whether you're heterozygous or homozygous? I don't know. We can't tell just by looking at you. So what do we have to do? We'd have to do a test cross. We do not do test crosses with people, okay? So don't worry about that. We won't do that experiment. So all that takes is we got to take you ladies and we got to cross you to a homozygous recessive individual. Again, this is why we don't do this experiment. So if we cross you to a homozygous recessive individual, we'll see what your genotype is. Actually, nowadays, though, of course, instead of genetics, we do it with molecular biology. We take a blood sample. We can actually screen for the presence of various mutant alleles. So we can do that without forcing you to have a baby, fortunately. Okay, so here's an example. What's the result of a cross between a plant with the yellow phenotype and a plant with a green phenotype? So those plants, assuming they're purebred, we're going to get an individual that's going to look like one of these two cases. If our yellow plant, our yellow seed, is homozygous, it's big G, and then we cross it to a homozygous recessive variety, that this is a test cross now. We do that, what we'll find is that all of the progeny are going to have the dominant phenotype. See my little Punnett square? Makes it obvious, right? What if, though, that yellow pea is heterozygous? It looks like this, big G, little g. The phenotype is still round. It's invisible to our eyes. And we cross that individual to a homozygous recessive one. Little g, right there. What you'll find when you look at your Punnett square is, okay, half of them, half the progeny will be big G, half the progeny will be little g. So half will be yellow and half will be green. Yay, you just determined what the genotype of that individual is. Of course, this only works if you can get lots of progeny out of it. Oh yeah, this is the real reason we don't do this with humans. If this were a human, of course, and you've got the dominant phenotype. Notice, both kinds of genotypes produce the dominant phenotype. It's just only half the time over there. So we'd have to have you have a dozen babies just to make sure. So to see, then we would expect 6 and 6. We could do a chi-square test or whatever probability you got and figure it out. So that doesn't work. But with things like pea plants and fruit flies, we can do this experiment. Okay, good place to stop. So Wednesday, exam will be released. We're going to expand our Mendelian perspective, and we're going to look at dihybrid crosses. We'll introduce Mendel's fourth postulate, and I'll show you a better way to calculate the outcomes than drawing Punnett squares. Okay, so we're still on chapter 3. We will get through chapter 3.